Welcome to lecture on Mendelian genetics and extended Mendelian genetics. Mendel figured out the laws of inheritance many decades before the discovery of DNA and the understanding of its structure and function. He worked with pea plants as he was able to clearly observe the 7 different traits and their expressions in the offsprings. The observable traits that he focused on were flower color, flower position, whether it's axial or terminal. Now these are the 2 positions. One is dominant, one is recessive. So each of these traits have one dominant and one recessive expression. Stem length is either long or short. Seed shape is round or wrinkled. Seed color is yellow or green. Pod shape, if it's inflated or constricted. And finally the pod color, yellow or green. Another reason that Mendel chose pea plants was due to distinct male and female organs present in each flower. This made self-fertilization and cross-fertilization easy, and he was enabled to control the traits. So now, let's look at some of the terms that he used before we start learning how to make Punnett squares. So in a typical breeding experiment, Mendel would cross-pollinate 2 contrasting true breeding pea varieties. So he would have homozygous tall plant cross with homozygous recessive plant. So that would be, if you have to represent it as big T, crossing with small t, and he called it the P generation, which is the parental. When the parental generation crossed, they produced the offsprings, and these offsprings were called the F1 generation. It was mainly Mendel's quantitative analysis of F2 plants that revealed that 2 fundamental principles of heredity, the law of segregation and law of independent assortment. What that means is he kept crossing these plants, and over time he realized that there are certain traits that express, and he called them dominant, and the others were masked by the dominant trait, and he called them recessive. So by the law of segregation, 2 alleles for a character are packaged into separate gametes. And this is what we now know is the process of meiosis. So if the blending model were correct, let's look at this. P generation that I just explained earlier. The purple flowers are dominant and white flowers are recessive. When they cross the F1 generation, all plants had flowers that were purple in color because capital P is dominant over lowercase p, which is white. So earlier I mentioned that his analysis were based on quantitative data. He calculated the number of plants. So for example, if Mendel recorded, let's say 705 pea plants, and purple flowered in F2 generation showed up to be 224, and white flowered F2 from the original cross were only 25% and 75% were purple, he realized that purple is expressing as homozygous as well as heterozygous, giving us the ratio of 3:1, which is also called the phenotypic ratio when you can see the physical traits, or genotypic ratio when you analyze the genetic composition of the traits. The genotypic ratio in this case would be 1:2:1. So when F1 generation crossed, we had 1 homozygous purple, which is capital P, 2 heterozygous, which is capital P, lowercase p, and 1 homozygous recessive, which is lowercase p. By the way, recessive will always be homozygous because you need 2 alleles to express the trait. So here is what I explained earlier. You can study these Punnett squares, and also I have given you the practice problems, so make sure you practice them. So before we move on, I have mentioned a lot of words. These are the vocab words that you should know. Character. Character is basically a feature that can be inherited. Trait is the variant for a character. True breeding, plants that self-pollinate, and every generation, subsequent generation, the same genes are transferred from parents to offspring. Monohybrid cross is a cross in which only one single character is crossed with the other single character from the other parent. P, or the parental generation, is the true breeding. F1 is the first filial generation. F2 is the second filial generation that is produced from crossing F1 crosses. More vocabulary, there's alleles, which is the alternate version of a gene. So if one gene is tall, what's the alternate or the allele for that? Would be either tall or short. Dominant allele is the one that's expressed. Recessive allele is the one that's not expressed in the presence of the dominant allele, but can only be expressed when there are two copies of that recessive allele. Homozygote is a pair of identical alleles for a character. So here's an example. Homozygous dominant would be capital B, and homozygous recessive is represented always by lowercase b and lowercase b in this case. Genotype is the genetic makeup, and phenotype is the physical appearance of the organism. So here you see once again the crosses all the way from parental generation to F2 generation. Please practice these problems in the packet that I've given you and understand how to do the monohybrid crosses. Now, moving on to test crosses. What are test crosses? Test crosses are designed to reveal the genotype of an organism. What does that mean? Now, this process cannot be carried out in humans. It can only be carried out in plants and in animals. Now, in plants, let's say you would like to know if the purple that's a dominant color in flower, is it homozygous dominant or heterozygous dominant? So what you do is you take that dominant phenotype and cross it with the recessive. And based upon the offsprings, you will figure out if the parent, the dominant parent, is whether homozygous or heterozygous. In this case, if you look at the first Punnett square, we are assuming that the purple flower is both homozygous dominant. So we have capital P gametes from one parent crossing with the lowercase p from the other parent. And as a result, all flowers in the progeny are purple. Therefore, that proves that the dominant parent is actually homozygous for this trait. On the other hand, if we assume that the purple is heterozygous, that means one allele is dominant and one is recessive, which are represented by capital P and lower p respectively, and we cross it with the lowercase from the other parent, then the 50% of the offsprings will be recessive and the other 50% are dominant, and we know for sure that our parent, purple parent flower, is heterozygous dominant, not homozygous dominant. So this is how you can determine by the simple process of crossing whether one organism is dominant or recessive for certain trait. These test crosses are done, they're practiced a lot in animal husbandry as well as in breeding processes. So let's talk about Mendelian inheritance and rules of probability. So basically the Punnett squares, in other words, are multiplication tables. And it tells you the probability of the event to occur. So you have already been calculating, I've talked about the ratios of 1:2:1 or 3:1. You heard those ratios in the lecture. Basically, that is the probability. 75% to 1, 25%, or 3:1 ratio is the chances in which certain traits can appear. Next, now we're going beyond Mendelian genetics. Let's talk about the incomplete dominance. And this is how the beautiful color in the nature appear. That's by the mixing of the colors. So in flowers, if red is dominant, the homozygous red is dominant. If you cross it with homozygous recessive white, you see the F1 generation is all pink. That means the heterozygote color is representing the intermediate between the red and white. Now, if you take the F1 generation and you cross two pink colors with each other, the F2 generation shows one homozygous dominant, two heterozygous pink, and one homozygous recessive. So in this case, the heterozygote is incompletely dominant. So it's bringing in the trait that's right in the middle of the dominant homozygous and the recessive homozygous. Codominance is where two alleles are expressed equally, and those two alleles both are dominant, and they do not interfere with each other. Their expression is co-expression, and their proteins do not mask each other. So the codominance, here's a problem that I've given you, and I would like you to pause at this slide and solve this problem, find the answer, and then move on to next slide. Another example of codominance is the blood groups, which is also an example of multiple alleles. So not all the alleles are either dominant or recessive. Some traits have more than two alleles. For example, our blood groups have O, A, B. These are the three alleles, and then we have the codominant allele, which is AB. We will talk more about this in our next lecture when we talk about the blood groups and multiple alleles. Now, moving through this quickly, poly means many, and genetic, genes. Many genes control one trait. So that is called polygenetic inheritance. As the organisms become more and more complex, there are many other ways that genes are expressed. So qualitative variation usually indicates polygenic inheritance. This occurs when there is an additive effect from two or more genes. A good example of polygenic inheritance is in humans, the height. Height is controlled not just by one gene or two alleles, it's controlled by many factors. And finally, we're going to talk about pleiotropy. Pleiotropy is the opposite of polygenic. Poly is many genes controlling one. In this case, one gene controls many traits, and a good example of pleiotropy is sickle cell anemia. In this case, one gene controls so many traits, and an abnormality in one gene can affect the organism in so many different ways. So this is where we're going to end this lecture. I will be talking more about this in our next lecture when we talk about human genetics. So if you have any questions, please feel free to call me, email me, or ask me in class, and I can elaborate more on this. Hope you enjoyed this lecture and hope you have taken good notes. Thank you.